Assalamualaikum and good morning. Today our group is gonna present our poster of proposal research that entitled Development of Sustainability Pest Management Strategies for Sarawak Black Pepper Piper using Trichoderma species. First of all, black pepper in Sarawak face major threat from fungal diseases like Phytophthora foot rot and blackberry. In order to prevent these diseases, our current reliance on chemical fungicide which raise cost and harm the environment and lead to resistance to pathogen. That's why we have choose Trichoderma species, which is beneficial fungi that are effective as a biocontrol agent. Some native species like T. harzenium, T. varians, and T. brevicompactum has shown strong effect against key pathogen. So our goal for this research is to discover and evaluate native Trichoderma species from Sarawak that can be effectively used as an eco-friendly biological control agent against fungal diseases in black pepper plant. So the objective of this research is to isolate and characterize native Trichoderma species from Sarawak and to evaluate their potential as biological control agents against major fungal pathogen affecting black pepper using morphological, molecular, and in vitro dual culture.
Next, I will be presenting the methodology parts, which consists of 4 main steps. This study begins with field survey on 5 black pepper farms in Belaga, Sarawak, where we collected samples from plants showing symptoms of foot rot, yellowing, and blackberry disease. In this step, plant tissues such as leaves, roots, stems, and berries were surface sterilized and cultured on PDA media to isolate the pathogenic fungi. The next step involved the isolation of Trichoderma species. Trichoderma were isolated from soil and tree bark sample collected in the biopark in surrounding forest in Bintulu. These samples were then cultured on Trichoderma selective medium or TSM, and pure isolates were obtained using a single spore isolation. In the next step, we use polyphasic approach for characterization, which include both morphological and molecular methods. Morphological identification was done based on colony growth patterns, color, conidiophore branching, and spore structure. On the other hand, molecular identification was done using DNA extract from 7 days-old cultures, which target the ITS region via PCR. The sequencing results were then analyzed and compared with NCBI GenBank for species confirmation. Next, to assess the biocontrol potential, we use a dual culture confrontation assay. Trichoderma isolates were co-cultured with 3 black pepper pathogens, including Fusarium solani, Coletrotrichum gliosporioides, and Phytophthora palmivora on PDA plates. Inhibition of micellar growth was measured, and the degree of antagonism was scored based on their invasion and sporulation. This approach allows us to evaluate the effectiveness of Trichoderma strain as a biocontrol agent for black pepper disease.
Next, for the result, we isolate 3 major pathogens, which is Fusarium solani, Psilotrichum thiosporioides, and Phytophthora palmivora. From the soil samples, Trichoderma harzenium was the most common species, which is 65%. In the dual culture test, T. harzenium and the T. tawa shows the strongest inhibitions up to 71% pathogens growth reduction. So for the discussion, T. harzenium was the strongest biocontrol potential and adapt well to the local condition, making it suitable for use in Sarawak culture. Morphology alone wasn't enough for identification, so we used DNA sequencing, which is for future work. We'll focus on greenhouse and field trial to test real-world effectiveness. For the conclusion, tihazonium was the most effective native biocontrol agent, with several isolations show strong inhibitions against key pathogens, proving that trichoderma can be a sustainable alternative to the chemical fungicides.